Welcome to solverfy.
Hi everyone, my name is Eric and let's review problem 47. So in this question, they are asking us to describe the process of molecular cloning. Now, molecular cloning is the process where you take your DNA of interest, you insert it in a plasmid, and then you're going to place it in a host to replicate and produce more amounts of your DNA of interest. And the process to do that is actually what we're looking for here. So we're looking at 4 different choices, A, B, C, and D. And I'll be walking us through what is the correct answer and why the other answers are not correct. So the first answer is letter A. So let's start with A. So they're saying that the foreign DNA and the plasmid are cut with the same restriction enzyme and the DNA is inserted into a gene. So let's start there. So our plasmid is a circular piece of DNA and it's mostly common found in the bacteria. So with the plasmid, we are going to cut at specific sites with the restriction enzyme, and that's going to allow us to insert our foreign DNA into the same spot. And in the problem, they described it as cutting into the LAC-Z gene. So by cutting into the LAC-Z gene, we are actually going to be disrupting this gene. And the LAC-Z gene is responsible for metabolizing lactose. So lactose is a molecule, and LAC-Z is the gene that allows the host to break down and utilize lactose. So that would be the first part where we're going to be inserting our DNA of interest inside the LAC-Z gene, right? And then they said that the DNA and the vector are annealing. So then that would allow us to make a new plasmid, but instead our DNA of interest will be bonded in the spot where the LAC-Z should be, right? And then this plasmid is going to be transferred into a bacterial host and it's going to be selected for using ampicillin, right? So somewhere on this plasmid will be an AMP-resistant gene, so that allows the bacteria to survive on ampicillin. So that's how we know if the plasmid has been taken up into the bacteria. The ones that survive on AMP resistance is what we're looking for. And once we have it into a bacterial host, we're going to be able to produce more plasmids with our DNA of interest. So it's going to replicate and produce our plasmid with our DNA of interest. And how we can tell that our DNA has been taken up into the correct spot is that if we introduce what is called xgal, xgal is similar to lactose. So xgal is actually the artificial version of lactose. So by introducing xgal, if the bacteria cannot metabolize xgal, then that means that our bacteria have successfully taken up the foreign DNA, and therefore our molecular cloning is successful. So part A, following those steps, is true. The answer would be part A. But to further explain, let's look at B, C, and D. So in B, already the first step is denaturing using high heat. So that is not correct. We don't denature. We have to restriction enzyme cut first. So B is not an answer. Let's look at C. So C, they're going to cut. Okay, so that's the good part. They're starting with cutting and they insert it into the plasmid. Then they allow it to anneal. But then the thing is here is that they said at the end here that it shows an inability to synthesize xgal. We're not synthesizing xgal. We're introducing xgal into the environment. We're giving it xgal so that we can see if it metabolizes it or not. So because we're not synthesizing xgal, C is not the correct answer either. And finally, let's look at D. So in part D, they are saying that they want to introduce it into a viral vector, and that is not part of molecular cloning because typically we're going to be focusing it in a bacterial host, right? And additionally, we're not synthesizing xgal. So that is also another reason why we are not picking answer D. So the process of molecular cloning, the answer is A. Thanks for watching, and I'll see you next time. Bye-bye.